Okay.
Thank you. Thank you for your kind introduction, Chair. Well, today we are going to, we have seen so many presentations about the biomaterials and stem cell, the materials which are for the modifications and the bioengineering of stem cell as well. But today, this time actually I'm going to talk differently that we are developing different types of biomaterials for gene therapy. So here, before we go further, we should know like, sorry. Well, so gene therapy is nothing but the modulation or correction of the wrong genes in based on the different pathophysiology of the diseases in the human body. So as we see, like in case of the drug delivery, firstly, generation one, which is a conventional drug delivery like tablet, capsule, liquid orals, and all those which have already a long back. And then generation two came as a second era where we can control the release of the drugs, the release of the active molecules from the matrix of the polymer. And then third generation is something which is very inspiring and one of the hot topic, that is the gene delivery system. So here we try to imply different kind of DNAs, RNAs, or transcription factors. Yeah, it doesn't work. No, it doesn't. No, it's okay. Well, so a collection of different types of approaches for the treatment of different types of human diseases that rely on the transfer of genetic material is nothing but a gene delivery system. So we can either try to cure these different types of genetic materials with the help of transferring genetic material like DNA, RNAs, or in case of the different disease like cancer, genetic disease, as well as different types of infectious disease. So if you can see the applications of gene therapy, there are a wide number of applications like inherited disease as well as acquired disease, different types of cardiovascular disorders, cancers, neurodegenerative disorders, as well as infectious disease, and so on. So these all diseases are like main target, and plenty of researchers are focusing on those research areas, either by viral vectors or different types of non-viral vectors to carry these genetic materials. So to go basically here, I would like to show that the basic mechanism of gene therapy, where anionic polymer, which is a DNA, can be generally complex with the help of cationic polymer, and then that ionic interacted complex can be then carried further toward the cell membrane, where the cell membrane itself is again a negatively charged. So we try to keep intact what the net charge of this polymer DNA complex in a bit positive way, so that it can easily get adsorbed on the negative charge cell surface, and then it undergo endocytosis, and then further the release from the endosome, and then later it shows its it releases the DNA genetic material, and then later it goes to nucleus. So here there are four major elements of the gene delivery, like targeted disease. Like it is very important, like what kind of disease we are targeting, what is the pathophysiology of the disease, what kind of a different kind of a genes which are responsible for that particular disease. For example, it's cancer, so what are different pathways, and so to carry that particular gene to the target site, that is what actually the gene carrier, which is what actually is very important. So we are focusing that particular area a lot. And then there is the gene expression. So after the delivering that particular gene, like how this gene will be expressed and how its physiological activities will affect or try to cure the disease. So here, as I said, like gene carrier is the one actually we are going to focus more. So there are two different types of gene carrier, like viral vectors as well as non-viral vectors. Non-viral carriers, non-viral. So until now, so many viral vectors have been researched until now, but there are so many disadvantages, something like carcinogenicity, immunogenicity, or insertional mutagenesis is a main problem. Also, self-replication by the viral vectors also is one of the big disadvantages. So these viral vectors such as retroviruses or lentiviruses, adenoviruses, adenovirus, all these kind of have some limitations. So that's why many researchers shifted their focus from non-viral to viral to non-viral vectors, such as polymers, lipids, or combination of the polymers and lipids like lipopolyplates or naked DNA, sonopolation, electroporation, calcification, all those. So here we are going to talk about mostly on these non-viral vectors such as polymers. But until now, we can see different types of clinical trials are being performed on the different types of vectors. You can see the maximum trials until now have been done mostly on the retroviruses. And retroviruses actually among the viruses, among all the viral carriers, those are the successful and efficient one. But again, there are so many limitations with this retroviruses because of their insertional mutagenesis, also the self-replication. So here, we can see the trials, different clinical trials on the gene therapy by different countries. And then we can see by type of a disease. So you can see cancer is again at top. I mean, there are millions of pathways to control cancer, so it is not very easy. So plenty of researchers are working on different types of cancer, like from the brain cancer until the all throughout the body. So that's why there are so much work of gene therapies going on in this cancer area. Then comes neurology as well. Then again the musculoskeletal problems and so many. So if you can see, depending on the disease, depending on the type of carrier, and also the type of cells which are being targeted mostly by the gene therapy. Here you can see this immunocells. The immunotherapy is one of the highest or the topmost area where most of the gene therapies are targeting right now, or the different types of MSC cells as well as the bone marrow cells or adipose cells as well. But among the immunocells is at top of the list, and then we can see how this immunity or the immunogenicity of the different cells can modulate, and we can try to cure those particular cancers with the help of this immunotherapy. There are these are the different key companies which are involved in the production of the gene therapy products, such as Oxford Biomedica, Genzyme, Transgene, or Planted Genetics, or also Veromed. So they employ different types of viruses, such as like retroviruses, or we can say like non-viral vectors. Among those, obviously polymers top the list, polymers or the lipids. But because of the toxicity of the lipids and also non-biodegradability of the lipids, again it has a limitation. So the polymer shows what gives a typical platform or the versatility, like we can modify those polymeric structures and in a different way work according to the disease requirement. And we can modify, we can decorate, we can give a cosmetic effect according to the different types of cellular barriers. So if you can see the barriers which actually this particular nanocarrier space, firstly the extracellular barriers, as we know that systemic delivery or reticular endothelial system, or different types of proteins available in the systemic circulation, stability. So those are all the barriers are very, very essential to be overcome by these nanocarriers to successfully deliver in the target site. The second part is the intracellular barrier, where actually once it goes, when the nanoparticles goes actually inside the cell, once it is endocytosed, after the endocytosis, during the endocytosis, it needs to be released quickly from the endosome. Most of the times it happens like the content, the cargo, actually remains in the endosome for longer time. It doesn't get released from the endosome. Because cell has its old function, the cycle, like early endosome, late endosome, and then finally if it doesn't come out of the endosome, then it goes to the lysosome and then finally degradation. So we try to employ such kind of polymers which allow this content or the cargo of the polymerase DNA complexes to come out of the endosome as much as possible, as quickly as possible, by employing different types of amine functional groups so that it can have a high buffering capacity, what we call like proton sponge effect. And then because of that, they come out of the endosome as much as possible during its acidic condition. And once it comes in acidic condition, it gets released in the cytosol, then ultimately it will come in contact with cytosol at physiological pH. The polymer will get degraded at that particular physiological pH, and then it will release the DNA content. So as I explained that these particular extracellular and intracellular barriers, like epithelial barriers as well as circulation and the blood components such as RBCs or different types of enzymes or serum proteins. Also this reticuloendothelial system, which is also one of the important systems where we have to combat while successfully delivering these nanoparticles. So we need such kind of an inert surface in order to have that successful carrier, successful overcoming these extracellular barriers. In case of intracellular barriers, as I said, like cell membrane is the first one. The cell membrane, crossing the cell membrane barrier, again the endosomal membrane, nuclear membrane. Also that particular DNA releasing at the right time is very, very important at right site, that location. So we need kind of specific carriers such as cationic or different types of multifunctional carriers which can have a specific activity so that it can successfully deliver at the target site. So if you can see the timeline of gene therapy until now, since this 1990, the first successfully reported gene therapy clinical trial at the French Anderson Center it was about the severe combined immunodeficiency syndrome, which was because of the deficiency of this adenosine deaminase enzyme, and that was the first successful trial was done. Then in 1999 there was another trial, but there was unfortunately a death of a patient, and that's why there were so many precautionary measures were being employed or imposed by the government authorities to ensure the safety of the gene therapy in a very effective way. So above this arrow, all these are the successful events of the gene therapy, even these also, except these two black boxes. And these are the deaths of the patients during the clinical trial. So that's why USFDA could kind of impose a strict regulations to ensure the safety of the materials or safety of the carriers for the gene therapy trial. So if you see specifically how these gene delivery considerations, so whatever the materials we develop for the gene therapy, it should be specific enough. It should target only a desired cells, not to any other cells or throughout the mast cell. It should have efficacy. So the potency is very, very important in order to have a proper therapeutic effect or clinical effect. So the delivery efficiency depends on the different disease requirements, like a cancer, or if it is a cardiovascular disease, if it is a neurodegenerative disorders, or depend. So what kind of a pathophysiology this disease have, we have to design or we have to develop our carrier in such a way that it should overcome all those barriers and deliver a genetic material successfully to the target site. And then finally comes the safety, which is very, very important, because safety, unless and until we ensure the safety measures, obviously no health authority will give approval for the further clinical trials as well as its launching in the market. So our objective was to use the polyethylene which is one of the golden standard polymer, which is very, very effective in case of the gene therapy. Actually, that polymer was initially used as in the paper pulp industry, but because of its two forms, like a linear and branch, it has a different types of chemistry, and then we can utilize that chemistry. So it can be given as a different, administered by different routes of administration, the animal study as well as the human study as well. But the problem with this polyethylene is that it interacts with the different blood components because of its high charge density as well as the amino groups. And that's the reason it has a non-degradability, and that's why it shows high toxicity. So higher the molecular weight of polyethylene, higher the toxicity. But if you have a low molecular weight of polyethylene, we can control the toxicity. So our objective was to synthesize different types of hyperbranch polyestyramines using the low molecular weight polyethylene. As I explained, that high molecular weight polyethylene is very highly toxic. So we try to use a low molecular weight polyethylene so that it will, our successful polyestyramine carriers will have a better water solubility, better DNA condensation, a similar way high buffering capacity as well as proton sponge effect, so that it can easily escape from the endosome and then release the content in the cytosol. It will have less toxicity because of the ester linkage, so that ester linkage can undergo hydrolysis at physiological pH, and it will have high transcription efficiency. So in the first part, we synthesize low molecular weight polyethylene and the copolymers from the low molecular weight polyethylene and the polycaprolactone, which is another FDA-approved molecule. So here, as we know, that polycaprolactone is a non-toxic hydrophobic and USFDA approved, and it's also biodegradable one. And this polyethylene, which is high transcription efficient polymer, it has a pronounced effect, but unfortunately, if we use a high molecular weight polyethylene molecular weight, it has high toxicity. So that's why we decided to use a low molecular weight polyethylene, which will have synthesis. This polyestyramine carrier will be biodegradable, high transcription efficient, and low toxic. So this is a scheme of the reaction here. We try to use 3 different types of the molecular weights of the polyethylene, 600, 1200, and 800 Dalton. And this polycaprolactone diacrylate is around 530 Dalton. So by michelation reaction, this particular polycaprolactone diacrylate was conjugated to this ethylenimine, and this is the representative structure of the polyestyramine. So here we can see the different types of ester linkage which are susceptible for hydrolysis at the aqueous pH, and it will ultimately have a low toxicity. So we have tried different weight ratios, feed ratios, and we have tried to synthesize a small library of almost 9 polymers using different feed ratios and the PI, PCl to PI, PI to PCl. And then among these, we selected only three polymers which are having a high or similar range molecular weight and also properly PI mole percent. So these are the biophysical properties of the polymer. Like it has, because of the polycaprolactone presence, it is a hydrophobic. So it has a long degradation. So that will be called a control or sustained release property. So when we check the degradation half-life, it was almost 4.5 days. So this is how actually this is such a long duration for the degradation. The particle size of these nanoparticles were almost less than 200 nanometer, which gives a better application for its interstellar delivery. Also, if you check the particle size, after particle size zeta potential, it shows almost 30 millivolt around net charge, which is sufficient enough to get a adsorb on the cell surface, which is negatively charged. And in this case, we also check the morphology of the particles. The particles were almost spherical enough so that it can show a better delivery efficiency. So firstly, we characterize these polymers by checking their toxicity in 3 different cell lines, such as 293, FG2, or Tella cell line. And in all these 3 cell lines, the polymer were very safe. I mean, even at high concentration, we can see here at different kind of NP ratio. Well, these NP ratios are nothing but like the nitrogen of the polymer to the phosphorus of the DNA, that is N to P ratio. So this is nothing but it is kind of a rational particular ratio where we can use high concentrations of the polymer in the further upcoming experiment. So to increase the concentration, something like, so here we can see like NP ratio 5, 10, 20, 30, so the polymer concentration goes on increasing. And even at high concentrations, we can see here, it doesn't show any kind of a toxic sign. So almost it shows like 80 to 70 to 80% cell viability. So that indicates that polymer can be used further for the in vivo experiment also. Then we check the transcription efficiency using the PGL3 as a reporter gene. So by luciferase assay, here we check the RLUs per milligram of the protein, and we can find that in case of 293 cells, FG2, or even Tella cells, all the 3 cell lines, polymer showed a much better transcription efficiency compared with this pink bar polyethylenimine 25 chloralone as a positive control, because that is the highest transcription efficient polymer, it's positive control. So polymer were much better than that, because generally what happens actually even at particular concentration, the PI shows high transcription efficiency, but later on it shows toxicity and then cells automatically get died, even though they get transcription. So it is not at all safe, but ultimately, on the other hand, polymers were safe enough and non-toxic and showing the high transcription efficiency. Also, when we check by using another reporter gene such as the GFP by flow cytometry, we can see here the polymer showed high transcription efficiency at NP ratio 20 and 30 compared with PR25 product. We also decided to use this material for in vivo application, so we decided to use the GFP as another reporter gene and distribute and administer it by aerosol administration to the mice. So after the sacrifice, after 2 days of the expression time, we can see here the polymer PCL-PI and PI. We can see the expression, particular green process expression higher in case of the polymer, indicating that it is a non-toxic. At the same time, it has a high transcription efficiency compared with the positive compared PI. So after getting this particular success in initial level, then we decided to have a specific targeting. So we decided to conjugate folic acid to this polymer with the help of polyethylene glycol as a linker, because we know that PG is one of the safe molecule and FDA approved. It kind of boosts up the stability of the polymeric materials in the system circulation and decreases the immunoreactions. So that's the reason actually we try to use this particular PG as a linker to conjugate the folic acid moiety to the polymer, so that it will have a receptor-mediated endocytosis, the folate receptor, because most of the tumors, the cancer tumors, they overexpress folic acid. So we try to conjugate this folic acid by activating the folic acid by disease genesis chemistry here. So this activated folic acid was then again added to the biofunction PG. So we have used this biofunction PG. Initially we used the 5000 Dalton, then later because of the transcription efficiency what was low, Then we decided to use a low molecular weight to like 2000 Dalton. So this 2000 Dalton bifunctional PG was then added to folate PG by this amide linkage. And then by using this chemistry, we added this polymer. So this is the final structure, folate PG, PCl, PI. So this is the folic acid moiety at the end, which is responsible. So generally minimum 5 to 10 millimole percent of folic acid conjugation is sufficient for the targeting folate receptors in cancer. So we checked for the toxicity in both the cell lines, the toxicity, the polymers were non-toxic at high concentrations. Then we checked the transfection efficiency in case of the HeLa Hg2 cell lines. So initially you can see in the first figure here you can see in case of the FPPA and PPA, like folate containing polymer and folate without folate polymer. So that is a notification here. So you can see with and without, we can see there is a difference. The folate containing polymer shows high transfection efficiency because of the high specificity for the folate receptor. So we decided to check the competition here, folate containing cells and non-folate containing cells. So Hg2 and Kg cells we can see here. In case of the Hg2, which is the hepatocellular carcinoma, there are much abundant amount of the folate receptors available. It is a lack of the folate receptor, but in case of the Kg cell, which is the oral carcinoma cell line, it has an abundant number of the folate receptor. And then we can see here high transfection efficiency of the polymer, indicating that it shows the receptor-mediated endocytosis. We also check, again, a competition assay that by blocking this folate receptor with the help of 200 nanomole of folic acid. So after blocking this particular folic acid, we can see here with and without folic acid, the transcription efficiency is different. So in case of the with folic acid here, the transcription efficiency gets suppressed. That indicates that the folate receptors were suppressed and blocked. And that's the reason they were not able to show this particular receptor-mediated endocytosis. And that's the reason we got a less transcription efficiency. So in this way, we try to use, in this same experiment, we try to use another TAM67 as a therapeutic gene. And then we delivered this particular therapeutic gene TAM67 to the xenograft mouse model. And then we have checked that number of the size of the tumors was reduced drastically. Then in the second part, we decided to use another sugar molecule, or the sugar alcohol-based polymers. Here, we try to use glycerol dimethacrylate because glycerol, as we know, that is a sugar alcohol, and it has free hydroxyl groups. So these free hydroxyl groups are responsible for better cellular mobilization, at the same time hyperosmotic effect, and ultimately it will lead to the cellular uptake. So this glycerol dimethacrylate was successfully synthesized. The polycermin was successfully synthesized using the glycerol dimethacrylate and the polyphenylene, which is a branch one, by Michael addition reaction. And this is a representative structure of the glycerol polymer, glycerol PI polymer, which we call PA, polyestyramine. So this polyestyramine was again characterized by NMR and different methods. Half-life, there is some particle size, or the zeta potential were successfully characterized, also the morphology by the TAM, and it shows like excellent properties. So we decided to apply it for further the toxicity. In different cell lines, HeLa Hg2 or even 293 cells, all the cell lines we can see, compared to this green bar of the PI25K, it shows very, very high cell viability, indicating that the presence of ester linkage in the backbone lead to the hydrolysis in the cytosol, and thereby there is no toxicity. Because generally toxicity arises from the blood, the deposition of these kind of non-biodegradable materials for the longer time in the cell, and ultimately it comes in contact with different serum proteins as well, or different cellular proteins, and that leads to toxicity. By checking the transcription efficiency by luciferase assay using the PGL3 as a reporter gene, you can see here all these three different cell lines, 293 or Hg or HeLa, polymers were high transcription efficient compared with PI chlordalton, and also compared with lipofectamine as another control. So that indicates the advantage of the ester linkage in the backbone of the polymer. We have to check this transcription by the flow cytometer, the FACS analysis. So after the confocal experiment, we can see here the difference between our polymer and the PI25K as a control. Polymers were like showing high fluorescence, especially the gene expression, which was being carried by the polymer to the medium PI. Obviously, compared to the PI, was much higher. So we further we decided to check the effect of serum. Since we try to employ these polymers by the maybe systemic circulation, we need to ensure whether these polymers are safe enough to travel or delivered by this systemic circulation. So we checked the effect of serum. So with and without serum, there was no much profound difference found. That indicates that those polymers can also be employed by the stem circulation or intracellular delivery in an effective way. Well, so we also checked, since this polymers contains 3 hydroxyl groups, we decided to check because it has a hyperosmotic effect. So to ensure the hyperosmotic effect, we just check the packed cell volume of these particular polymers in the 290 cells with the glycerol 3, 4, 5, and 10% as another control. And it shows that almost a kind of a similar. So if you can see with the negative control, which is without glycerol and with glycerol, that indicates that how this packed cell volume is getting decreased. That indicates that like packed cell volume is lower. That indicates that the particles or the nanoparticles containing these polymers can be uptaken by the cell membrane or cells in an effective way because of the hyperosmotic effect. Another effect here we can see that bacillomycin A1, which is a specific emitter of the bacterial type of proton ATPase, because these amine-containing polymers, they actually follow this particular proton sponge effect. So to ensure that whether our polymer really follows that pathway or not, we block this particular proton ATPase with the help of this external bacillomycin 200 nanomole. And then after blocking, we can see the difference that with and without bacillomycin. With bacillomycin, the transcription suppresses drastically, almost 50%. That indicates that proton sponge or the bacterial type of proton ATPase has been blocked by the bacillomycin. So that indicates that polymer follows that particular, or carrier follows that particular pathway, which is of the proton sponge effect. And this proton sponge effect was discovered by J.P. Behr from France in 1995, almost 30 years. So this is how actually it was successfully, we successfully synthesized these carriers. After the aerosol administration of the similar polymer by the bulk C mice here, we have checked after the 2 days the gene expression, and we can see the difference here. The polymers showing high gene expression compared with the control and P25 chloraltone also. So the third part, another similar way we another synthesize the glycerol triacylates in a similar way.
I'm afraid that is.
Oh yeah. How much time?
Because there are only 4 minutes. So the next section we have.
We are fully prepare the next session.
So
how much time
maybe
now?
within 2 minutes.
Okay, sure. Thank you. Okay, so the last part I would like to show here, the lactitol, another sugar alcohol. So this lactitol, because of the free hydroxyl groups, it has a hyperosmotic effect, which generally, And in this case, we try to use the sugar alcohol polymers for the hepatocyte targeting, for the liver cancer, because as we know that parenchymatous cells of the liver contains the glycoprotein receptors. So these particular glycoprotein receptors can be targeted by this particular sugar alcohols or sugar alcohol moieties. So here we decided to synthesize this polylactidol-based polymer and the PI copolymer, so that it will have a specific or receptor-based endocytosis property, but by targeting this hepatocyte. So we have synthesized this lactitol in a successful way, and then we apply for this. So here we can see this particular first part, which is carrier uptake part. Since because of the free hydroxyl groups, it has a high uptake because of the hyperosmotic property. The similar way, the ester linkage here you can see, which is having a biodegradation part, and then finally the amine-containing amine group, and that is specifically for the DNA binding part. So this was successfully characterized by different spectroscopic techniques as well as the cell viability technique by MTSSA. Also, we checked the transcription of this particular polymer in Hep-G2 cell lines, and also with the help of the competition assay, we can see here, it shows that particularly this polymer has a specificity for hepatocyte cell line, or the hepatocytes or Hep-G2 cell line. So in order to have this particular competition assay, we block this glycoprotein receptors by the external galactose concentrations, and we can see whether really this polymer follows the pathway by the glycoprotein receptor or not. And then we have got the positive results here that by blocking this particular galactose receptors or the glycoprotein receptors by galactose concentration, the transcription has gone down. So it indicates that receptor endocytosis pathway was profoundly used. So by using this polymer for the biodistribution, so we conjugated this Cy 5.5 molecular probe, and then deliver the material for the biodistribution by the telemetry injection. So we have found that almost highest distribution in case of the liver was abundantly found. That is the specificity of this particular polymer for the cellular receptors and the liver. So further, we use this polymer. So I'll just escape this one, and then I'll just go to the in vivo data that this was successfully used for the liver cancer, lung cancer here also. This in another way. So here we use the OPA1 gene, and then OPA1 gene was successfully silenced. So by looking all this particular data, we can see that we try to use different types of materials and try to synthesize a safe and non-toxic polymer, which actually have high transcription efficiency and also a better cellular property. So these are our collaborators, and those we're very thankful for our study as well. This is our group who actually work for this particular all this experimental research work. So thank you for your attention and your question.
So it's a very interesting speeches, but due to the time limitation, I'm afraid that we cannot ask any question. If you are interested, so please go outside and discuss.
Okay.